Let's imagine that this is the nucleus of a cell located in the pancreas. It is going to start the process of making insulin. The nucleus holds DNA, which kind of looks something like that. I am not a very good drawer. The DNA is packaged by special proteins to form chromatin, you see over here. And then that undergoes further condensation to form chromosomes. Chromosomes contain our genes, which altogether these all hold our genetic code to make different proteins and other molecules to be sent throughout the body. RNA, not to be confused with DNA, can match up with the DNA too. Sorry, that's not very good drawing, but it copies the instructions to make insulin. Okay, let's just... After the RNA copies the instructions, you can see the nuclear envelope here. It has these small pores, and those pores allow the RNA to leave the nucleus and then joins with ribosomes. These little dots located on the rough endoplasmic reticulum are ribosomes. The joining of the ribosomes is where the beginning of the synthesizing of the protein occurs. Let's just note that ribosomes can either float out free, so somewhere like out here, if that was the rest of the cell, in the cytoplasm, or they can be attached, as you see here, to the rough ER. There is also smooth endoplasmic reticulum, which does not contain any ribosomes on the outside. The proteins are then sent to a small temporary sac made of membrane called a vesicle. You see that here. Vesicles' main function is to ferry biological molecules to various regions throughout the cell, so other organelles. The vesicle in this case has transported the protein to this structure located over here called the Golgi apparatus. This is where a specialized set of membranes act as a finishing room, so the product is packaged and exported to the proper place. Here is just a full picture of a cell to kind of understand the whole process. So again, this is our nucleus. That is where the DNA occurs in the start of our insulin. When the RNA copies the instructions, it moves out through the pores to this endoplasmic reticulum, to the ribosome specifically, where the proteins are created. Then vesicles transport to the structure. This would be our Golgi apparatus, where the finishing product is packaged and shipped to the correct part to use the insulin. And that is a synopsis of how insulin is made from a pancreas cell.